Okay, so we continue on the second part, which is more to characterize the association between different types of objects. So typically two objects, you have red, green ones, and you want to know whether or not, for example, the green are close to the red. And what does it mean? So for the rest of the presentation, the term coupling design some spatial proximity between objects and a proximity that is significant. So quite naturally, you can define a cross function K. So instead of counting for each point the number of points around, you're going to, for example, to consider the red one, and you're going to count for each red point and a parameter R, how many green ones you have. You're going to list for all the red, you sum on all the red, you count how many green you have, and you still have the same boundary correction and the same prefactor. And again, you can look at the quantiles and the percentage and distance of coupling of association. So how many, so for example, if you have this type, how many green points are associated, are close to red, and how many are just randomly distributed. And this is not trivial, again, because even if you have like spots in a domain, and especially if you have a lot, and if R for a given R, you will still have a lot of, you can have a lot of neighbors, but this proximity is not, can be non-significant because just you have a lot of molecules in a bounded domain. And this is the illustration here. I mean, in the subway, this is normal to be close to a lot of people, and it's much less natural in an empty space. So in imaging, in multiplex imaging, usually you have a lot of pre-processing, data pre-processing, where I'm going to extract the different objects. So here, for example, this is a two-color image of synaptic molecules. So this is just a fluorescent imaging. And you have a lot of algorithm in image analysis to segment, extract these objects, and go into a mathematical formalism where the objects, the spots, have a position, but also a mark. So this is why here we call this type of processes marked point processes. The point is a position, and the mark can be the shape, the color, not the position. Usually the position is in the point process. So here again, this is the typical formalism that we have. And if you look on internet or in textbooks, mark point processes, this is a collection of points in the domain with mark. So the mark here is typically the color or the size. And then you can use all the tools of spatial statistics, and especially the replace function, to characterize the position of a given mark compared to another one. I just mentioned other methods to look at the coupling, and especially if you use like a cluster analysis, then you can look typically at cluster overlap. So here, this is a paper based on Voronoi tessellation. So you can define some cluster. So I recall that the clusters, they are region with a small adjacent Voronoi cell. So here in purple and green. And you can look at the what is the proportion of overlap between clusters. And this is another method. Another method where, again, they look at local cluster and look at the overlap between local clusters. I don't exactly remember the method they used to cluster data. So yeah, as I mentioned, we build method based again on the Ripley scale function, which is nice because you can characterize statistically the distribution of the function for complete spatial randomness again. And here it's a little bit different from the other one because instead of looking at a given position R, we look in concentric rings around, because then you can characterize the distribution of points in each of these rings and specify which ring, in which ring you have a statistical accumulation of red molecules here. And again, a boundary correction. And yeah, I have like 3-4 slides to show how you can, how do you know, how you can define the rings that contain too many objects, meaning that there is some statistical, some significant coupling between the two distributions. And again, the hypothesis here is not that the two distributions are completely independent. So it's a little bit, it's a relaxed hypothesis stating that the number of red objects in the ring is proportional to the area of the ring. So meaning that the red are spatially distributed. You have no hypothesis on the green, but the hypothesis that the red, let's say that the red are randomly distributed in the domain. And actually, so the expected number of objects in each ring is going to be Gaussian if you have a sufficient number of objects. So let's say that we have a sufficient number, and so you need the mean and variance to characterize the function in each ring. So the mean is exists. This is the area of the ring as before, and for the variance, it's a little bit more tricky because it's going to depend on the density, the number of spots around. But also, again, you have a boundary correction, which complicates a lot the analysis. And here, you're going to have some interaction between green objects. So when greens are very close, you see that their annulus, their rings or annulus, they're going to overlap. And so you have to account for this, especially in the variance, to correct for this, because typically, if you have random, so let's say that by chance you have a lot of spots here, and let's say that these two spots are very close together, you want to mitigate a bit the fact that by chance you have a lot of red one here. You don't want to say twice that you have a lot in this ring and in this ring, because the green one are very close. So there is an interaction between the different rings that need to be taken into account. So this is like a small explanation, but when you do the derivation, you can see that the mean is clearly like the area of the annulus. And for the variance, so again, you will have a boundary correction. So here it's a bit complicated. And here, some intersection between the different annulus that is going to be included in the term of variance. So that's now you have the mean, we have the variance. So now we can again define a centered variable with zero mean and unit variance. So you remove the mean G, you divide by sigma, square root of variance, and if the red ones are randomly distributed, this tends to normal distribution. And again, if red are just randomly distributed, you expect to have some Gaussian vectors where each component is just normally distributed. And so that's good, because then you can, for example, build a statistical test. You can consider the max value, the sup, and the sup, if you have n Gaussian variable, what is the probability that this sup is above this value. This is 1 minus the probability that all the components are under this value. And because all the components are just randomly distributed, you have that the p-value of your observed diagram is 1 minus this. So this is a cumulative density, cumulative function for the normal distribution, sub, and at the power here, because you have to invert the fact that the probability that the sup is above a given value is 1 minus the probability that each component is below this value. So you can build a p-value, but most importantly, you can, so I skip this, but actually there is a universal threshold for a Gaussian vector. And if you're interested, I can give you the reference. So they've computed that the best threshold to identify which components are large and which one are non-significant is square root of 2 log n. And where n is the number of n. Why do you need a threshold that depends on n, the number of components? It's because imagine that you have a lot of components. If you have a lot of components, by chance you will have some components that are high or low. This is just natural. If you have like 1,000 components, 1,000 rings, you will have rings that can reach 3, 4 just by chance. And so you need a threshold that adapt to n, and actually there is a lot of literature on this, because when people have decided to compress images, they used some transform based on wavelet, and they developed some statistic to select the significant component to encode the image and remove the noise. And actually, you can show that the distribution of the wavelet coefficient for noise is just Gaussian. And so they have developed a lot of statistics to select the significant component in a Gaussian vector like this. And this is the same threshold, and then you can use this to select the component, so the rings where there is an accumulation that is significant. And then you can, because you know for each ring, if you have a significant number and how much objects you have, you can then define a probability for each couple to be for each. So for example, for these two, there is a probability that they are associated, which is how many objects I have in these rings divided by how many I expect. So this is here. So this is G0i, this is the number of objects. G0i plus mu I, I'm a bit lost. So here, this is a condition that you are going to consider only the ring that are above the threshold. So you don't want to get information from these rings which are noise. And in this ring, you are going to look at, yes, because there is 0, because I have removed the mean. But if I add the mean, this is the total number of molecules I have in the ring. And here, this is the number above the expected number for spatial randomness. So here, this is the excedent number of molecules in the ring, and it gives me the probability that each of these objects is associated or not. So a bit technical, but the take-home message is that you can define which rings are in which rings you have a significant accumulation of objects, and based on this, you can define the distance of association, because then you can accumulate all the spatially associated objects around all the rings. And this is a small example. So we performed some spatial analysis on 3 color images of synaptic molecules with Lydia Danglot, who is working at the Institute of Psychiatry and Neuroscience in Paris. So here she did a 3-color imaging with synapsin, a pre-synaptic molecule in green, PSD95, a post-synaptic molecule in red, and Homer in blue. And we could segment, identify all the spots in an entire neuron. And we ran this on 10 neurons, so more than 30,000 spots, and we could identify single molecules that are coupled to only one. So here, this is a synaptic Homer, synaptic PSD, or extra-synaptic coupled PSD Homer. And most of the synapse contains the three, the synapsin, the PSD, and the Homer, and we could identify the size and the distance between all the molecules with nanometer precision. So that was nice. And I'll conclude this analysis of spatial association with complex shape objects, because here we have only considered points. And we have also developed some methods using level sets to deal with complex shape or tissue or anything. So the idea is that instead of looking at this concentric distance around points, we map the domain in a level set. So meaning that for each point in the domain, it is at a given distance from the closest contour of an object. And so, for example, here, this point is at a distance from one of these contours. And so you have a merge. When you are going to grow the level set, look at the distance, then it's going to map all the domain. And this is quite interesting because first, you don't have any more boundary correction. And second, this term of interaction, which is really a problem when you have a lot of molecules and a lot of coupling, you have no more, because then you have only one single level set that is going to map for each point the position to green objects in general. You don't consider individual green objects, but you just say them as an ensemble, and you look at the distance of each red object to the ensemble of green ones. And it is important, this is just a simulation that we did, where, let me just remember, so we have red, and I'm colorblind, so it's very difficult for me. So here, I think we have red points, red objects. And around these red objects, we have blue points that are associated to the red objects at some distance, and green ones that are randomly distributed. And what we did was to grow the size of the red. So here, red are points. Here they are a little bit bigger, and much bigger. So here at the end, we are looking at these objects with some complex shape and big. And we are looking around with some level sets, and we look at the accumulation of maybe the accumulation of the green and not the blue. I mean, maybe the blue are random and the green are accumulated at some distance from the objects. And we compare, so we simulate a given percentage of accumulation. So what is the percentage of my blue-green objects that are associated to the red? So 0 to 0.6. What is the association I measure? So here, this is just a code name. So Pasteur, this is a method using level set mapping, and SODA for statistical object distance analysis. This is the method I presented before, where you have concentric rings around. So it works pretty well for points. Both methods works pretty well for points, but you see that when the objects becomes bigger and bigger, Spatial Path, so the method with level set, is working very well in all cases, while for the method using concentric rings, when the objects are not point, but they have some size and some shape, then it's going to either overestimate or underestimate. So it's not good. And this type of method, this level set method, is particularly important in digital pathology because you can again image. Okay, it's a bit blurry, but you can image whole tissue. So this is a biopsy for a patient with non-small lung cancer, quite aggressive type of cancer. So and we have, so here this is like so this is the whole tumor, and inside the tumor we look at the response, the immune response to the tumor. And we looked at the distribution of the different immune cells in cyan and around the contour of the tumor. So here you need really to get a level set mapping of this, and in between the cells, in between cells. And so there is a first step where you detect cells. So here it's not easy. We use some deep learning detection process that gives us the different, for example, the mast cells, which is one type of immune cells, and the T cells in yellow. And here we can also segment manually or automatically the border of the TER, which is tumor epithelial region. So let's say tumor. And then we can use the level set to map, for example, around mast cells in cyan, the accumulation of yellow, around yellow, the accumulation of cyan, around the tumor, the accumulation of cyan, and again here. So we have like 4, and it works pretty well. And now I would like to conclude with, okay, so this was an example with 2 to 3 colors, but what happens with a lot more of color, of type of objects? And this is becoming increasingly important because of technological innovations. So here, for example, in fluorescence microscopy, there's Raman imaging. So don't ask me a lot of details about Raman imaging because it's quite complicated, but let's say that you can image a lot of colors, a lot of different molecules inside a cell. Or here, this is multispectral imaging. So this is something where you can divide finely and image up to 6 colors inside the cell. So you are using a lot of different labeling, fluorescence labeling, and then you can go up to 6 colors and here even more. So, and these data sets are growing. So we have much more than 2 colors. As I mentioned before, we have also this type of application in where the immunohistochemistry imaging. So you use different label to color in 2, 3, 4, 6 colors the tissue. And this is not that big because this is one example, but there is now 3 or 4 different technologies where you can image. So for example, here for the codex imaging, you can image. So you take your sample, your biopsy, then you use different antigen with N. So N typically here is around 50. And so you put your antigen on your samples, and actually you then use 3 antibodies per 3 antibodies. So all the antigen, the antigen is going to bind to a specific molecules. And then you are using, so for example, 3 antibodies that are going to bind to 3 antigen, so 3 molecules. You image, you wash, you put 3 new antibodies on your sample to image 3 other molecules you wash, you do this like, I don't know, 20 times, for actually 3, 3, 3, 3. And at the end, you have a tile of images that each contain 3 colors, 3 colors, 3 colors, and then you can combine all the images and take 50 colors. So the only thing right now is that this type of imaging is still quite complex and not widely spread, but it becomes more and more distributed. And this type of, and then you can obtain this type of multicolor images. So here, for example, again, I'm colorblind, but you have like 1, 2. So this is just an example of 1, 2, 3, 4, 7 color. And so you see that the tissue, you can have like cells, small structures, cells, or longer structures, etc. So you need multiplex, meaning that you have a lot of different type of cells and structures. You need level sets, clearly, because all the structures are not just reduced to points. And last example, this is something, a paper that we did with, that got published today with David. So here we use quite standard labeling. I think this is, again, immunohistochemistry with few colors, but you can detect all the cells. So here, this is microglia. But using the morphology and the local neighborhood of the cell, you can use deep learning to classify the cell. And so then for each detected cell, you can classify it as one type of microglia. So at the end, we had 5 types: proliferative, amoeboid, cluster, phagocytic, ramified, with quite a good what is the name? A confusion matrix. So you have this input, you train a unit with some manual annotation, and then it's going to give a probability for each pixel in the segmented mask to belong to one type of cell. And so at the end, you look at the total probability for one segmented structure, and you decide whether it is an amoeboid, a cluster, a phagocytic cell. And so you have a threshold. But you can also keep the probability information and say that this structure has a probability of 50% to be proliferative, and 30% to be amoeboid, and 20% to be cluster. We have also derived some statistics to account for these probabilities. And this is the type at the end of tissue imaging that you have. And, so you see that here clearly you have a big structures that look like points, and then you can clearly analyze, for example, the accumulation of one type to another, etc. And so I have 3 more slides because I wanted to discuss actually the challenge, because now that we have this multiplex data sets, what do we do? Because right now we can look at the association between 2 types, 1 to 2, 1 to 1, 1 to 3, 2 to 3, etc. And actually now there is a new direction of research directly looking at multicolor spatial relations, 2, 3, 4, and identify this type of clusters. But this needs the development of new statistics to look at multicolor assemblage. And also something that is becoming increasingly clear, especially in tissues, is the fact that so you can look at the local relations between the different cells or the cells to structure, but actually this is like local, but you can also have information. So here this is labeling of different cells, and here this is the first clustering of the different region of the tissue in a subregion. And actually it becomes increasingly clear also that, for example, the relation between this supersized region 6 and 2, or 9 and 2, etc. So relation between regions themselves and the localization of cells inside this neighborhood is important to decide whether or not, for example, here the cancer is of stage 1, 2, 3, 4. So for doing the classification and to predict, for example, the survival of a patient, it becomes increasingly clear that you need the local association, but also the semi-local association, for example, what is the relation between this neighborhood and how you define this neighborhood, it's not clear, and both. And so this also needs the development of new methods, or to integrate this multi-scale information. And last but not least, so you have like multiplex imaging, so multicolor. You have this multi-scale. But even if you look at one-to-one relation, so for example, as I mentioned before, the T cell, one type of cell to another, vice versa, and the accumulation of 1 and 2, even by looking at this 4 association, if you take, so these are 8 patients, A1 to A8, and for each patient, you can look at this Ripley analysis, and you see that actually you have a lot of variability between the patients, and still you see like conserved patterns. So for example, here there is an accumulation of mast cells around T cells. Here also, here you have clearly a repulsion. So the T cells, they are far from the tumor, which is not the case for the mast cells. So you see conserved pattern, but clearly you see a variability. And how do you account for this? So you have a lot of information because you have where there is accumulation, how much, etc. So how did you consider this information into some deep learning methods to say, okay, this patient, the cancer is very aggressive, the immune response doesn't work. And this one, the immune response is working perfectly. So, and this has some implications because, for example, if you give some immunotherapy to a patient, it costs a lot. So thousands of euros for a treatment based on immunotherapy. So at some point, you want to know whether or not the patient responds to the treatment, needs the treatment, or needs an adaptation of the treatment. So, quite important question. And that's it. Thank you. Thank you.
Okay, thanks, Thibault. Great.